Welcome back, everybody. Ready to dive into some more fascinating microbiology?
Always.
Today, we're going to be tackling how antibiotics can actually exploit the folate pathway to, stop bacteria. We'll be looking closely at Chapter 8 from Antibiotics, Challenges, Mechanisms, Opportunities by Walsh and Wensfitz.
It's a classic.
Yeah,
It really gets into the details of how some of these antibiotics work.
It does, and I think we might find some surprises along the way.
Oh, I think so too.
When I think of folate, I usually think of leafy greens or prenatal vitamins.
Right.
Not really, bacterial warfare.
Yeah, understandable. But folate actually plays a much bigger role than just that.
Okay.
It's almost like a formaldehyde sponge, which is really important for a lot of different reactions in the cell.
A formaldehyde sponge?
Yeah.
Okay, I did not see that coming.
And a lot of that ties back to how DNA and RNA are different from each other.
Right, they have different sugars.
Exactly. DNA uses deoxyribose to make it more stable. And then there's the classic base swap. You've got thymine in DNA instead of uracil in RNA.
Yeah.
And that need for thymine for the DTTP building block, that's what the bacteria are really vulnerable to.
So how does the formaldehyde sponge fit into all of this?
Well, bacteria have this enzyme called thymidylate synthase.
Okay.
And it needs methylene tetrahydrofolate, which is a folate derivative to work.
So that's the connection.
Yeah, methylene tetrahydrofolate is carrying this one carbon unit
Right.
that becomes part of the thymine. And this is really neat. The CH3 group in thymine actually gets its third hydrogen directly from the folate.
Oh, wow.
Yeah.
So it's not a simple one-step process, I'm guessing?
You're right. It's a 3-enzyme cycle that's constantly going to make enough DTMP for DNA synthesis, serine transhydroxymethylase,
Okay.
thymidylate synthase, and dihydrofolate reductase.
Wow.
All working together.
And each of those is a potential target.
Oh, absolutely. The fact that bacteria make their own folate.
Unlike us.
Exactly. That makes this pathway a great target for drugs.
So let's talk about some of these antibiotics.
Okay.
I know sulfonamides, like the sulfa drugs,
Right.
have been used for a really long time.
A long time.
How did they disrupt this folate process?
They're kind of like masters of disguise. They look like PABA, which is a crucial ingredient for bacterial folate synthesis.
Okay.
And they bind to this enzyme called dihydropteroate synthase.
Right.
And they basically trick the bacteria into making a useless folate-like compound.
So no DNA production.
Exactly.
But I'm sure that bacteria have found ways around this by now.
Oh, they definitely have. That's kind of the arms race that we always see.
Yeah.
But thankfully, we've got another tool, trimethoprim.
Yes, cotrimoxazole.
Yes.
So we hit them with both.
Exactly. Sulfonamides target dihydropteroate synthase.
Right.
And trimethoprim goes after dihydrofolate reductase, which is really important for recycling dihydrofolate back into tetrahydrofolate to keep that folate cycle going.
So with both drugs,
We create a double blockade.
Yeah.
And that's why cotrimoxazole is so powerful.
Keeps them on their toes.
Yes. It's harder for bacteria to become resistant when we use both.
That makes sense. But what about newer discoveries? Anything else that targets this pathway in different ways?
There is Abysomycin C.
Abysomycin C. That sounds like something out of a sci-fi book.
It kind of is. It was discovered in the Sea of Japan.
Okay. Tell me more.
It's got a very unique way of working.
Yep.
It targets PABA synthase, which makes PABA that we were talking about earlier.
So it goes after the very first step.
Exactly, before the folate cycle even starts.
Wow, that's amazing.
And there's another interesting thing.
Okay.
The most potent form is actually its atroposomer
Tetroposomer.
called atropabysomycin C. This molecule shuts down PABA synthase for good. It forms a covalent bond with the enzyme.
A covalent bond. Wow.
Yeah.
That's pretty permanent.
It is. And the term atroposomer, it rings a bell.
Vaguely. From organic chemistry.
Ah.
Remind me what that is again.
Basically, they're stereoisomers that can't really switch between forms.
Okay.
Like they're mirror images of each other, but they can't easily interconvert.
Gotcha. So with abysbosine C, it's this very specific atropisomer that has just the right shape to really lock on to PABA synthase.
You got it.
This is so cool.
It is.
I'm learning so much about how bacteria survive and how we can stop them.
It really is a constant back and forth, but understanding these pathways and how antibiotics work can help us stay one step ahead.
Well said. I think it's time to put your knowledge to the test.
Okay.
First question. What are the two major differences between DNA and RNA?
A classic but important one.
All right. Question 2. Explain how the folate coenzyme helps make DTMP. Think about that one carbon unit we talked about.
Right.
Question 3. Describe how sulfonamide antibiotics actually work.
Those sneaky molecular mimics.
Exactly. Question 4. Why is it better to use sulfamethoxazole and trimethoprim together as cotrimoxazole rather than just one? And finally, question 5. How does abysomycin C stop bacterial growth? Remember that deep sea discovery?
This one should really test your knowledge.
Okay, pencils down. Ready to go over the answer.
Absolutely. Let's see what we've learned. So that first question about DNA and RNA,
Right.
they have different sugars.
Yep.
DNA has deoxyribose and RNA has ribose. And that missing oxygen in deoxyribose
Right.
makes DNA more stable.
It's got to last longer.
Exactly. And then there's the base difference.
Yeah.
DNA uses thymine.
And RNA uses uracil.
Right. They both pair with adenine.
Right.
But that little difference between thymine and uracil really makes a big difference.
It does.
And that's what some antibiotics take advantage of, that need for thymine or DTMP.
Okay, so question 2 was all about the folate coenzyme.
Right. That folate coenzyme carries a 1-carbon unit, and that ends up as the methyl group on thymine.
Like a little taxi.
Yeah. And without it, thymidylate synthase can't do its job.
And no DTMP.
And then no DNA.
Right. So then we talked about sulfonamides.
Yes.
How do they actually work?
So they look a lot like PABA,
Right.
which bacteria need to make folate.
Okay.
And they bind to dihydropteroate synthase, which is the enzyme that uses PABA to make folate. But because they're not exactly the same, they stop the enzyme from working, right?
So bacteria can't make folate.
Exactly. And that's a big problem for them.
Because we can just get it from food.
Yeah, we don't have to make it.
So smart. Then question 4 was about cotrimoxazole.
Yeah, that's a good one.
I never really understood why we use both drugs together.
It's all about hitting the pathway in 2 places.
Okay.
You've got sulfamethoxazole, which is a sulfonamide,
Right.
and trimethoprim, which blocks dihydrofolate reductase.
So it's harder for resistance to develop.
Exactly. It's much more effective than using either drug on its own.
That makes a lot of sense. And finally, abysomycin C.
Ah, yes. From the depths of the sea.
So cool. It targets PABA synthase.
Right, that's the first enzyme in the whole pathway.
And it uses that atropisomer.
Abysomycin C.
Yeah, which can form a covalent bond with the enzyme.
Exactly, a permanent bond.
Amazing. And humans don't even have that enzyme.
Nope. So it's a really good target for antibiotics.
This has been a great deep dive.
It has. It really shows how much we're learning about bacteria.
And how to stop them.
Exactly. And we need to keep learning because they're always changing.
So true. We've learned so much about this pathway.
We really have, but there's always more to learn.
Oh, definitely. Are there any other areas of research that you're excited about?
Lots. Researchers are working on ways to make the antibiotics we have even better.
Okay.
Like finding new ways to deliver them so they can get past resistance mechanisms.
So we can use what we already have more effectively.
Exactly. And there's also more research on combination therapies.
Like cotrimoxazole.
Right. Targeting multiple steps at the same time.
Makes sense. What about the challenges of developing new antibiotics?
One of the biggest challenges is making sure they only target bacteria.
Right? We don't want them to hurt our own cells.
Exactly.
Yeah.
And we also have to think about the microbiome.
Oh, yeah.
Yeah.
All those good bacteria that live in and on us.
Right. Antibiotics can mess up that balance.
Yeah. So we need to be careful.
We do. Researchers are trying to develop antibiotics that only target specific bad bacteria.
So they don't harm the good ones.
Yes. And of course, there's always antibiotic resistance.
That's a big one.
It is. Bacteria are very good at adapting.
So we always have to be one step ahead.
We do. We need to keep tracking resistance and finding new ways to fight it.
It's a constant battle.
It is, but it's an important one.
Absolutely. Well, thank you so much for sharing your knowledge with us today.
It's been my pleasure.
This deep dive into the folate pathway in antibiotics has been fascinating.
I agree. It's a really important topic.
And if anyone wants to learn more, be sure to check out Chapter 8 of Antibiotics, Challenges, Mechanisms, Opportunities by Walsh and Wentzvitz.
It's a great resource.
Okay, before we go, let's do a quick quiz.
Sounds fun.
Question 1. Why is the folate pathway a good target for antibiotics?
Think about why these antibiotics are so effective.
Question 2. How is trimethoprim different from sulfonamides in how it works?
Remember, they each have a different target.
Question 3. What's an atroposomer, and why is it important for abysomycin C?
Think back to our discussion about those mirror images.
Question 4. Why is cotrimoxazole better than using either drug alone?
Think about synergy.
And last question. What are some of the challenges we face in developing new antibiotics that work on the folate pathway?
This one really makes you think about the big picture.
Okay, That's all the questions. We'll post the answers in the show notes.
Let us know if you have any other questions.
And until next time, keep exploring the amazing world of microbes.